Welcome to Chapter 10 on the muscle tissue. So there are three main types of muscular tissue that we're going to discuss. We're going to spend most of the time in these lectures talking about skeletal muscle, and then we'll kind of come back later and talk about visceral and smooth muscle, or visceral and cardiac muscle, excuse me. So we've got a nice little table here looking at comparing and contrasting three types of muscles. Skeletal muscle is obviously going to be located in association with the skeleton, and functionally it's going to be used to move bones. This type of muscle tissue is going to be multinucleated, meaning there are multiple nuclei per cell, and it's also going to be striated. So this means that there are going to be visible bands of light and dark in these cells. This type of tissue, skeletal muscle tissue, is also controlled voluntarily. This means it takes conscious effort to elicit some sort of response. The next type of muscle tissue that we have is our cardiac muscle. Obviously, this one is associated with the heart, and it is used to pump blood. These cells are typically multinucleated or single nucleated, one nucleus. They are also going to have striations, just like we saw in the skeletal tissue. And one unique characteristic of cardiac tissue is going to be the presence of these intercalated discs. Cardiac tissue is going to be controlled involuntarily, meaning that we do not have to have conscious thought in order for this muscle tissue to contract and relax. All right. This tissue is considered to be autorhythmic, and it's controlled. This autorhythmicity is controlled by hormones and neurotransmitters that are going to be responsible for regulating the speed and strength of the contractions. Next, we have the visceral or the smooth muscle organs, or the smooth muscle tissue. Excuse me. This is going to be associated with a variety of different organs, especially those that are associated with the GI tract, really important for peristalsis, for like the movement of nutrients and foodstuffs through the GI tract. This tissue is going to have one nucleus and is not going to be striated, so this is going to be a characteristic that's going to set it apart from the other two muscle types of muscle tissue that we talked about. And as we mentioned, this type of tissue, this smooth muscle tissue, is also involuntarily controlled. So looking at the various functions, the overall just general functions of muscular tissue. All right, there are going to be four main ones. All right, the first is pretty obvious. It's producing body movements. So this is going to be movement of the whole body, like walking around, as well as localized movements, like movements of foodstuffs through the GI tract. Right? It can also, functionally, muscular tissue can also work to stabilize body position. So skeletal muscle contractions are going to help to stabilize joints, as well as helping to maintain body position, such as standing or sitting. So this kind of helps you stay in an upright position. Muscular tissue is also going to be very important for storing and mobilizing substances within the body. So this storage is accomplished by sustained contractions of ring-like bands of smooth muscle, and these are called sphincters. And these are going to prevent the outflow of the contents of a hollow organ. So obviously this is going to be really important when it comes to things like holding your bladder, so you're not just urinating all over yourself at inopportune times. Also things like keeping food in the stomach so that it can begin that digestion process. All of that is going to be achieved by these sphincters. And then we also have, as far as our last function of muscular tissue, is going to be the generation of heat. So as muscular tissue contracts, it's going to produce heat. And this process of heat production is called thermogenesis. This is going to be when involuntary contractions in skeletal muscle, like known as shivering, this can also increase the rate of heat production. So it's kind of a handy sort of little homeostatic measure there as well. Next, we have the properties of the muscular tissue. So a big characteristic of muscular tissue is the fact that it is electrically excitable. And this simply means that it has the ability to respond to certain stimuli by producing electrical signals. And these signals are called action potentials, sometimes called impulses. So there are two main types of stimuli that are going to trigger action potentials. One is autorhythmic electrical signals, and these are going to arise from the muscular tissue itself. This is going to be things like the pacemaker, the heart's pacemaker, that sets the rhythm for the heart to contract. The other is going to be chemical stimuli. This is things like neurotransmitters that are released by neurons or hormones that are distributed by the blood. It can even be something as simple as a change in the local pH can trigger this action potentials. Our next property or characteristic for muscular tissue is contractibility. All right? So this is the ability of muscle tissue to contract forcefully when stimulated by an action potential. So when a skeletal muscle contracts, it's going to generate tension or force of contraction. This is done while pulling on its attachment points. This allows things to produce movement, essentially. We then have extensibility and elasticity, and these two are important, and make sure that you don't get them mixed up because there's a big difference here. So extensibility is the ability of muscular tissue to stretch within limits without being damaged. And then elasticity is the ability for muscular tissue to return to its original length and shape after the contraction or the extension. So it's important to know the difference between these two, between extensibility and elasticity. All right, extensibility is the ability to stretch. Elasticity is the ability to recoil or return to original length. So let's take a look now at the skeletal muscle tissue. So this is kind of interesting, and I didn't know this before I started teaching this class actually, that it's that each skeletal muscle fiber is going to arise during embryonic development, and this is going to develop from the fusion of hundreds or more of these small mesodermal cells called myoblasts. So each mature skeletal muscle fiber is going to have 100 or more nuclei, and once this fusion has occurred, the muscle fiber loses its ability to undergo cell division. So the number of skeletal muscle fibers is going to be set before you're even born. And most of these cells can and should hopefully last for most of your lifetime. So that's very interesting that they don't undergo cell division anymore. So basically, the number that you're born with is the number that you end with, ideally. They can obviously become bigger. So like if you go to the gym and get all buffed up, then yes, obviously you're not growing more muscle cells, but you're strengthening and enlarging the muscle cells that you have already. So it's kind of an interesting little side bit. So let's look then at the connective tissue components of muscular tissue. So this is going to surround and kind of encase the various parts of the muscle. So the subcutaneous layer is the first layer that we're going to discuss, and this is going to separate muscle from skin, and it's composed of areolar connective tissue and adipose tissue. It's going to provide a pathway for nerves, blood vessels, and lymphatic vessels to enter and exit the muscles. The adipose tissue of the subcutaneous layer is also going to be great for storing most of the body's triglycerides, and it's going to serve as an insulating layer that reduces heat loss. And it's also going to help to protect the muscles from any kind of physical trauma. So it's a great little cushion. Next we have the epimysium. This is an outer layer, and it's going to encircle the entire muscle. All right. It's going to consist of dense, irregular connective tissue. So here we can see our epimysium covering the entire muscle there. Our next layer is the perimysium. You can see this here, and this is going to be a layer of dense, irregular connective tissue, but it's going to surround groups of 10 to 12 or 10 to 100 or more muscle fibers, and this is going to separate them into little bundles that are called fascicles. Next, we have the endomysium, and this is going to penetrate the interior of each fascicle, and it's going to separate individual muscle fibers from each other. So the endomysium is mostly going to be made up of reticular fibers. And then next we have the fascia, and this is going to be a sheet or a band of fibrous connective tissue that is going to be deep to the skin and is going to surround the muscle, as well as some of the other organs in the body. So if the connective tissue layer extends beyond the muscle to form a rope-like structure, it's called a tendon. And if they form a flat sheet, then they're going to be called an aponeuroses. And we'll look at some of these in the lab when we get there today or later. Next, we're going to look at the microscopic anatomy of the muscle. So all the little tiny bits that actually make up the muscle. All right. So first we have the sarcolemma. All right. This is basically just going to be the plasma membrane of the muscle cell. All right. And then next we have the sarcoplasm, and this is the the muscle cell's cytoplasm, and it's going to contain a large amount of glycogen for energy production, as well as myoglobin for oxygen storage. We then have the transverse tubules, or the T tubules. These are going to be tiny little invaginations in the, which you can see here, kind of this sort of blue line. Right? These are going to be little tiny invaginations into the sarcolemma, and they are going to help to quickly spread muscle action potentials to all parts of the muscle fiber. Next, we have the myofibrils. These are going to be small thread-like structures within the sarcolemma, and these are going to act as contractile organelles in the skeletal muscle. The sarcoplasmic reticulum is going to encircle each of these myofibrils, and it's going to be similar to the smooth endoplasmic reticulum in non-muscle cells. Then our terminal cisterns are going to be these little dilated sacs that you can see here, kind of in the yellow, right? These are going to be dilated sacs of the sarcoplasmic reticulum, and these are going to butt up against the T tubules on either sides. So a transverse tubule and the two terminal cisterns on either side of it are going to form a triad. And in a relaxed muscle fiber, the sarcoplasmic reticulum is going to store calcium ions. The release of these calcium ions from those terminal cisterns of the sarcoplasmic reticulum is what's going to ultimately trigger that muscular contraction. So that whole setup is very important as far as actually having some functioning muscle there. Next, looking at the filaments that are associated with the muscle. So within the myofibrils, there are going to be these small protein structures called filaments. There are two main types of filaments. We've got our thin filaments and our thick filaments. Thin filaments are composed of actin, while thick filaments are made up of myosin. Both types of filaments are going to be directly involved in the contractile process, and there are going to be two thin filaments for every one thick filament in the region of filamental overlap, where those filaments kind of lay on top of each other. The filaments inside the myofibrils do not extend the entire length of the muscle fiber, however. Instead, what they do is they're going to be arranged in little compartments called sarcomeres. And these are going to be the basic functional units of our myofibril. So looking at the components of the sarcomere, we're going to go through all of these different parts of the sarcomere, and then we're going to look at an actual image of the sarcomere, and you can maybe try and put those two things together. So for starters, the components of a sarcomere are going to be organized into a variety of bands and zones. So the narrow plate-shaped region of dense protein materials called a Z-disc is going to separate one sarcomere from the next. So a sarcomere is going to extend from one Z-disc to the next Z-disc. The darker middle part of the sarcomere is going to be the A band, and this is going to extend the entire length of the thick filament. And then towards each end of the A band is going to be the zone of overlap, and this is where the thick and thin filaments are going to lie side by side. The I band is going to be lighter and is going to be less dense than a less dense area that is going to contain the rest of the thin filaments but no thick filaments. And a Z-disc is going to pass through the center of each of these I bands. The alternating dark A bands, however, are going to so the alternating between these dark A bands and the light I bands are going to help to create the striations that we see so typically in skeletal muscle fibers. So you can see these striations in both myofibrils as well as in the whole skeletal and cardiac muscle fibers. So a narrow H zone in the center of each A band is going to contain thick filaments but not thin filaments. And then supporting proteins are going to then hold the thick filaments together at the center of the H zone, and these are going to form the M line. And they're named this because they are going to be in the middle of the sarcomere. So M for middle. So here we have an actual image to look at all of the things that we just talked about. So we've got our Z discs, right, that are going to mark the borders of our sarcomere here. Right, we've got our A band and our, where'd it go? Our I band. Oops, sorry, technical trouble. Our A band and our I band here, right? We've got the H zone, so this zone of overlap, or excuse me, that's going to have thick but not thin filaments. Our I band only has thin, and the M line is going to be running down the middle here. All right. So here we can see like a little bit more of a zoomed in view of these protein filaments, and we'll actually look at these in just a minute. So here we have a very lovely table describing all of the various components of the sarcomere. Right? I would definitely recommend taking note of this table. Myofibrils are built from three kinds of proteins. We've got the contractile proteins. These are the ones that are going to generate the force during contractions. We've got regulatory proteins. These are going to help to switch between the contraction process on and off, to switch between contraction and relaxation. And then our structural proteins, and these are going to keep the thick and the thin filaments in the proper alignment, and they're going to give the myofibril elasticity and extensibility. And it's also going to be linked to myofibrils to the sarcolemma and the extracellular matrix of the cell. So starting with our contractile proteins, we've got two contractile proteins, myosin and actin. Right? And these are going to make up the thick and thin filaments. Myosin is going to be the main component of the thick filament, and it's going to function as a motor protein in all three types of muscle tissue. Motor proteins are going to pull on various cellular structures, and this is going to allow them to achieve movement by converting chemical energy in ATP to mechanical energy of motion. And this is going to be how we produce force. So in skeletal muscles, there are going to be about 300 molecules of myosin that are going to form a single thick filament. And you can see them kind of here. So each myosin molecule is going to be shaped like two golf clubs that are sort of twisted together. The myosin tails, which are going to be like the handle part of the golf club, are going to point towards the M line at the center of our sarcomere. And the tails of neighboring myosin molecules are going to lie parallel to one another and form the shaft of the thick filament. The two projections of each of the myosin molecules, the golf club kind of head part, are going to be called the myosin heads. So each myosin head is going to have two binding sites, and these are going to be really important for the whole process of contraction. So these two binding sites are going to be, one is going to be for an actin binding site, and the other is going to be an ATP binding site. So this is where the ATP binding site is going to be used to function as an ATPase, which is an enzyme that's going to hydrolyze ATP into ADP plus 1-phosphate, and this is going to be how we're generating energy for muscle contraction. The heads are going to project outward from the shaft in a sort of spiraled fashion, which you can sort of see here. And each head is going to extend towards one of the 6 thin filaments that are going to surround each thick filament. Our next contractile protein is the actin filament. This is what's going to be making up our thin filaments. And individual actin molecules, which we can kind of see here, they look like kind of like beads, are going to join together to form actin filaments that are going to be twisted together into this sort of helix shape. So they're sort of twisted. You can see that. So on each actin molecule, there's going to be a myosin binding site, and this is going to be where that myosin head is going to attach to those actin molecules, so that it can kind of walk down that thin filament there. Next, we have our regulatory proteins, and these are going to be really important to allow the contraction process to happen. So there's going to be a smaller amount of these two regulatory proteins that are called tropomyosin and troponin, all right? And these are going to be associated with the thin filaments. So in a relaxed muscle, the myosin is going to be blocked from binding to the actin because these strands of tropomyosin, which we can see here, these kind of brown strands, is our tropomyosin. And these are going to basically just cover the myosin binding sites on the actin molecules. These troponin molecules or strands are going to be held in place by our troponin molecules. So we've got these kind of holding them in place. So, however, when calcium ions are present, like when we're triggering a muscle contraction, they're going to bind to this troponin molecule. So troponin is going to undergo a conformational change, and this is going to pull that tropomyosin molecule, or that away from the myosin binding site on the actin molecule. And this is going to allow myosin heads to bind to the actin molecules, and the muscle contraction is subsequently going to begin. So that's kind of how that works. So those are two very important proteins there. We then have our structural proteins. These are proteins that are going to help keep the thick and the thin filaments of our myofibrils in proper alignment. And they're going to give myofibrils their elasticity and extensibility. And they're also going to link the myofibrils to the sarcolemma and the extracellular matrix. So our first structural protein that we're going to discuss is called titin. This is a huge protein. I think this is actually the largest protein that we have in the human body. It's gigantic. If you ever meet a scientist that studies titin, you should feel bad for them, because it is a pain in the butt to study because it is so huge. Hence the name, titin. So this is going to be a structural protein that is going to connect Z discs to the M line of the sarcomere. And this is thereby going to help to stabilize the thick filament positions. So overall, it's going to help a sarcomere return to its resting length after a muscle has contracted or it's been stretched. Our next structural protein is alpha-actinin. And this is a structural protein of the Z disc, and this is what's going to bind actin molecules of the thin filaments and the titin molecules together. Next, we have our myomesin. This is a structural protein that's going to form the M line of the sarcomeres, and it's also going to bind titin molecules, and it's going to connect adjacent thick filaments to one another. Next, we have our nebulin, and this is going to be a structural protein that wraps around the entire length of each of the thin filaments, and it's going to help to maintain the alignment of the thin filaments within that sarcomere. And then last but not least, we have our dystrophin. So this is going to be a structural protein that's really important in linking the thin filaments of the sarcomere to the integral membrane proteins in the sarcomere, in the sarcolemma. These are going to These are attached in turn then to proteins in the connective tissue matrix that are going to surround the muscle fibers. So in short, basically, dystrophin is going to reinforce the sarcolemma, and it's going to help it transmit the tension that's generated by the sarcolemma to the tendons. So if you have a mutation in this protein dystrophin, you could potentially have something called muscular dystrophy, and this is going to cause a significant weakening or tearing in the sarcolemma of these muscle cells, because they don't have that reinforcement that's needed to help to transmit that tension that's generated by the sarcomeres on the tendons, or to the tendons. So here we have, for your studying convenience, we have a little table summarizing all the various skeletal muscle fiber proteins. It's sort of a handy thing to look at. And then next we're going to talk about the contraction and relaxation of the skeletal muscle fibers. This is a really actually interesting topic, and a very interesting kind of set of sequence of events. So the first mechanism we're going to look at when we talk about contraction and relaxation of our skeletal muscle fibers is going to be the sliding filament mechanism. The muscle contractions are going to occur because those myosin heads are going to attach to and walk, quote fingers, along the thin filaments at both ends of the sarcomere. And this is going to progressively pull those thin filaments towards the M line. The myosin is then going to pull on the actin, and this is going to cause those thin filaments to slide inward. So consequently then, our Z discs are going to move towards each other, and the sarcomeres are going to shorten. So thanks to the structural proteins that we've just mentioned, this is going to allow for a transmission of force through the entire muscle, and this is going to result in the whole muscle contracting. So it's important to remember that changes in or it's important to like see these changes in the I band and the H zone as these muscles contract, right? So we can see here our I band, our H zone, and our A band. So we can see how these change significantly as that muscle fibers contract. So it's really kind of interesting to look at. We then have the contraction cycle. All right, this is going to be how essentially everything is going to bind, and this contraction is going to be able to occur. So it's all going to start with ATP hydrolysis. This is where the myosin head is going to hydrolyze ATP, and it's going to become energized. And this is going to allow it to be oriented so that it is able to bind to those actin filaments. All right. Then we have attachment. All right, and this is when the myosin head is going to bind to that actin filament. You can see that here. And this is going to form this cross bridge where these two filaments are connected, or are bound together. We then have our power stroke, which sounds pretty badass. This is going to be when the myosin head is going to pivot, and it's going to pull the thin filaments past those thick filaments, and then towards the center of that sarcomere. And then the ADP that's bound to our myosin fiber there, our myosin head, is going to be released from that myosin head. All right, this is going to be released, right, and then this is going to allow for detachment to occur. So as the myosin binds to ATP, again, right, so we release our ADP, we bind up some ATP, and this is going to cause the cross bridge to detach from the actin. And then the whole process is going to start over, where that myosin head is going to then hydrolyze this newly attached ATP, right, and then allowing for the whole process to continue. It's very cyclical. So if you have a copy of my slides, this link will be in there, and it just is basically showing a motor protein doing the walking along that microtubule. It's a very interesting, it's a funny little, it's a very funny visual to kind of look at. And we understand really why it's called walking down the filaments, because it really does look like it's sort of walking. It's very goofy looking. So next we have our excitation and contraction coupling. So this is going to be really important because this is what's going to stimulate this whole contraction to occur, with the excitation of these muscular cells and that triggering and that connection between the contraction. All right, so during relaxation, the levels of calcium are going to be fairly low in the sarcoplasm. All right, and this is because the calcium ions are being pumped into the sarcoplasmic reticulum, and this is being achieved by a calcium ATPase pump. So a muscle action potential then is going to propagate along a transverse tubule, and this is going to cause those voltage-gated calcium channels to undergo a conformational change, and these are going to open those calcium channels and allow calcium to flow flow out of the sarcoplasmic reticulum and into the sarcoplasm. And this is going to trigger the beginning of that contraction. So this concept is going to connect the events of a muscle contraction or a muscle action potential with a sliding filament mechanism. So it's putting two things together, our action potential and then the corresponding sliding filament mechanism, putting those two things together and connecting them really. So before we talk about that, we need to discuss the relationship between the tension in the muscle and the length of the muscle, and how this is going to relate to the contraction ability. The force of a muscle contraction is going to depend on the length of the sarcomere in a muscle prior to its contraction. So at a sarcomere length of somewhere between 2 and 2.5 micrometers, which is pretty close to the resting length of most muscle tissues, the zone of overlap in each sarcomere is going to be at the optimal range. So this is where at this range, the most tension is going to be able to be generated. So our maximum tension, or 100% tension, is going to occur when the zone of overlap between a thick and a thin filament is going to extend from the edge of the H zone to one end of the thick filament. And as the sarcomeres of a muscle fiber are going to stretch, they're going to become longer in length. And this is going to cause the zone of overlap to shorten. So this means that fewer myosin of those myosin heads are going to be able to contact with the thin filaments. And this is going to result in the tension of the fiber that the fibers can produce being significantly decreased. And we can kind of see that here. So as we stretch, as we increase the length of our fiber, we have a significant decrease in the amount of tension that's able to be generated because we're shortening that zone of overlap there. So when a skeletal muscle fiber is stretched to 170% of its optimal length, there's going to be no overlap that's going to occur between those thick and thin filaments. And because there's no overlap, right, none of those little myosin heads are able to bind to the thin filaments, and the muscle fibers are not going to be able to contract. So we're not going to generate any tension at all. On the other hand there, as the sarcomere length becomes increasingly short, so as we're shortening or kind of scrunching our muscle fiber there, this is also going to result in a decrease in the amount of tension that can be developed. This is going to be simply because the thick filaments are going to basically crumple and get squashed as they're being compressed by those Z discs. And this again is going to result in fewer of those myosin heads that are able to make contact with the thin filaments. So as a result, they're not going to be able to generate that tension. So normally a resting muscle fiber length is going to be held very close to the optimum range by those firm attachments of skeletal muscle fibers to bone via their tendons, as well as to other inelastic sort of tissues. So it's going to be, your body's doing its best to keep it within the optimal range so that contraction can be produced. Now on to the neuromuscular junction. Right? So this is where we're going to kind of join the nervous system to the muscular system. This is going to be how we're going to electrically trigger these muscle contractions. So for starters, we have something called a somatic motor neuron. And this is a neuron that is going to stimulate skeletal muscle fibers, causing them to contract. So a muscle fiber is going to contract in response to one or more of these action potentials that's going to propagate along its sarcolemma and through its system of T-tubules. Muscle action potentials are going to arise at a neuromuscular junction. Right? So the joining or the junction between the neuron and the muscular part of the whole system here. So the synapse between a somatic motor neuron and the skeletal muscle fiber itself are going to be what we're going to call this neuromuscular junction. A synapse is basically just a region where communication is going to occur between either two neurons or between a neuron and its target cell. And in this case, it's going to be between a somatic motor neuron and a muscle fiber. So at most synapses, there's going to be a little small gap, right? Because the nerve is not actually going to come in contact completely with the actual muscle. There's going to be something called a synaptic cleft. And this is going to separate those two cells by just a teeny tiny little gap. So because these two cells are not physically touching, the action potential can't actually jump that gap between those two cells. So instead, the first cell is going to communicate with the second cell by releasing chemical messengers. And we call these things neurotransmitters. So at the neuromuscular junction, at the end of our motor neuron, there's going to be something called the axon terminal. And this is going to divide into a cluster of synaptic cluster of synaptic end bulbs. And this is going to be the neural part of our neuromuscular junction. So suspended in the cytosol within each of these synaptic end bulbs are going to be hundreds of these little membrane enclosed sacs called synaptic vesicles. And these vesicles, these synaptic vesicles, are going to be responsible for holding on to and carrying acetylcholine. And this is going to be the neurotransmitter that's released at the neuromuscular junction. So the region of the sarcolemma that's opposite the synaptic end bulb is called the motor end plate. And this is the muscular part of our neuromuscular junction. So within each motor end plate, there are going to be between 30 million and 40 million of these acetylcholine receptors. And these are going to be integral membrane or transmembrane proteins. And these are also considered ligand-gated ion channels. And these are going to specifically bind to our acetylcholine. So these receptors are going to be very abundant in the junctional folds. And these are just basically deep grooves in the muscle end plates that are going to provide a large surface area for that acetylcholine to be able to bind. So a neuromuscular junction is therefore going to include all of the synaptic end bulbs on one side of the synaptic cleft, plus the synaptic cleft itself, and all of the motor end plates of the muscle fibers on the other side. So now let's look at the events that are going to occur at the neuromuscular junction in order for that are going to result in this production of an action potential. So for starters, we have the release of acetylcholine. Our voltage-gated calcium channels in a neuron's synaptic end bulb are going to open as a result of some sort of voltage change from our neuronal action potential there. And this is going to result in an influx of calcium. This in turn is going to cause the exocytosis of the neurotransmitter, which is acetylcholine. And this is going to result in the emptying of our acetylcholine into our synaptic cleft here. All right. We then are going to have that acetylcholine is going to traverse that synaptic cleft, and it's synaptic cleft, and it's going to bind to and activate our acetylcholine receptors. Acetylcholine is going to bind to those ligand-gated sodium channels that are on the motor end plate of the muscle, and this is going to cause an influx of sodium ions into the muscle. This in turn is going to result in the production of a muscle action potential. So this depolarization of the muscle and the resulting release in calcium ions from our sarcoplasmic reticulum is in turn going to trigger our action potential there. We're then going to have the termination of our acetylcholine activity, where this acetylcholine is going to get broken down by an enzyme called acetylcholinesterase. So it's going to stop triggering that action potential there. So then here we have this really great little summary image here looking at just an overall graphical depiction of the summary between the contraction and the relaxation of a skeletal muscle. Right? So we have our nerve action potential is going to propagate down our somatic motor neuron, and this is going to trigger the release of acetylcholine. Acetylcholine is going to bind to these receptors on our motor end plate, and this is going to trigger our action potential in the muscle. Acetylcholine is then going to get broken down by acetylcholinesterase that's been released from our somatic motor neuron. All right. So from there then, our action potential is going to travel along the transverse tubules, and this is going to trigger a change in the voltage-gated calcium channels. And this is going to cause the calcium channels to release that calcium it's going to release those calcium into that sarcoplasmic reticulum. Right? This is in turn going to, those calcium ions are going to bind to the troponin that we have that's on the thin filaments, and it's going to expose those myosin binding sites that are found on the actin. Then this is going to allow for contraction to occur when those myosin heads are going to bind to that actin, and they're going to do that power stroke and release cycle that we've just mentioned, and the filaments are going to be pulled towards the center of the sarcomere. All right. From there, then, the calcium that's released, those calcium release channels are going to close, and the calcium ATPase pumps are going to make use of ATP to restore the levels of calcium to lower levels within that sarcoplasm. Right? This, in turn, is then going to cause those tropomyosin molecules to slide back into position, where they're then going to block the myosin binding sites on that actin. Right? And then the muscle at this point is going to be relaxed. So that is all we have for this first lecture on muscle tissue. So if you have any questions, comments, or concerns, please feel free to contact me.